So we'll be moving on to Nick Nastasi from Ohio State University, and his talk is on the interaction of lunar dust, of dust on International Space Station, and on the growth habits in space habitats. Nicholas, are you there for us?
Yes, can you hear me?
Yes. Go ahead.
Okay, okay, great. Well, hi everyone. My name is Nicholas Nastasi. Currently, I'm a PhD student at The Ohio State University, and I am very excited to present for the first time at this Lunar Surface Science Workshop. And excited to discuss the interaction of lunar dust and International Space Station dust on microbial growth in space habitats. This is our current research, and we'd like to share some future research ideas. Next slide, please. So we know that dust is an important source of human exposure, particularly through inhalation. This is true on Earth and indoor environments, and is also true on spacecraft. So we know that any human occupied space will have dust, microbes, and moisture that are just naturally occurring depending on the occupants in it and what is going on in the surrounding environment. And previous generations of spacecraft, such as Mir and the Space Shuttle and currently the ISS, have taught us a lot about controlling microbial growth in these environments. However, it's still possible to get unintended growth to occur. For example, these images on the left are from the space station, a fabric panel where a moist towel was placed, and some fungal growth occurred on this panel. And also with the crops that are being grown on the space station, there's water involved with that too. And also you could see some fungal growth on this as well. Next slide, please. So we also know that dust composition affects microbial communities. And this study here that the figure is showing is Earth-based dust. And it's looking at several different rooms from one facility. You can see these rooms on the x-axis here. And it's showing how the fungal communities varied with the dust composition in each of these rooms. So each room had a different type of dust with different things in it. And you can see by these box plots here that the communities change depending on the type of dust that was collected. And this is important when we start to think about the mixing of lunar dust and dust that will be in the cabin of a space habitat. Next slide, please. So since we know this is a potential source of human exposure for the astronauts that'll be living in a habitat, it's good to know what type of dust will be present in the lunar habitat. So the first is this ISS type dust. The figure on the left here shows a ISS vacuum bag that was returned to Earth. This dust is primarily made of fibers from their clothing or the fabrics of the spacecraft material. Also a lot of human-generated materials such as skin cell and a lot of hair, and also potentially any chemicals and microbes that are present in the environment will settle and be absorbed onto the dust here. And also, of course, on the lunar habitat, we'll have lunar dust, which is this mineral-based dust, and it's very angular and jagged and has been notoriously difficult to remove. So we need to think about what happens when these two things combine in a occupied habitat. Next slide, please. So I want to talk about our current work, which is investigating how microbial growth in ISS dust is affected by relative humidity conditions. So we got 4 vacuum bags from the International Space Station. This dust was collected by vacuuming the HEPA filter covers on the ISS air ventilation system. They were brought down to Earth, where we did the study. You can see the figure here shows what the dust looks like when it comes out of the bag. And basically, we incubated this dust at varying relative humidity conditions in these jars right here, where we can control the humidity and the temperature. And after each incubation, we quantified the total microbial growth using qPCR. And we also wanted to visualize this using scanning electron microscopy. Next slide, please. So the first experiment we did was looking at fungal growth with varying relative humidity conditions. So these were all 2-week incubations at 25 degrees Celsius. At each varying relative humidity conditions shown on the x-axis here. So we went from 50 all the way to 100%. We also looked at the original dust. So this was dust without any incubation. So what was originally in it when we first received the bag. And the y-axis here shows the fungal concentration after these 2-week incubations. And we could see that fungal growth increases as relative humidity increases. These yellow bars indicate a statistically significant difference between the relative humidity shown and the original dust. So basically at elevated relative humidity conditions, which is considered anything above 80%, we do see this significant fungal growth compared to that original dust. Next slide, please. So another experiment we did was modeling this growth using a time of wetness framework. And we can model this even if the relative humidity is fluctuating. So basically this model, the time of wetness refers to the fraction of time per day that it is above that relative humidity condition of 80%. So we did this by varying 50 and 85%, and also 50 and 100%, which corresponds to elevated and saturated relative humidity conditions. So for the saturated or 100% relative humidity condition, this green bar shows a deactivation limited growth model. And this means that there is very rapid fungal growth at lower time of wetness, and it eventually plateaus as that time of wetness increases. On the other hand, for the saturated or the 85% relative humidity conditions, we have this activation limited growth model, which means that it takes some time for the fungi to adjust. So it's slowly growing. And then as time of wetness increases, we start to see this more rapid exponential growth. Next slide, please. So we also saw visual changes in the ISS dust after incubating at these elevated relative humidity conditions. On the left side here, you'll see what happened after incubation at 50% relative humidity. The top figure shows like an actual photo of the dust clump, and the SEM image right below that shows the fibers that make up the ISS dust. This is primarily the fabric from the clothing or from other panels and materials on the spacecraft. And then we could see at 100% on the right side here, with the naked eye, you can see the change in this.
Nick,
Yes,
excuse me, Nick, you have about 3 minutes.
sure. At 100%, you could see this visual change. It is darker, and you can actually see some fungal colonies indicated by these black specks. And through SEM, we can actually see the fungal structures on these fibers that make up the ISS dust, including hyphae, spores, and fruiting bodies, all of which indicate that fungal reproduction is happening. Next slide, please. It is important to note that this relationship between dust, relative humidity, and fungi is also seen in residential homes on Earth. So on the left here, this is the ISS dust, basically the material I just presented to you. And on the right here, we have Earth-based dust and carpet, which is something our lab also works on. And there are similar trends in fungal growth as relative humidity increases. And the time of wetness models varied slightly, and we're still looking to figure out exactly why that is. Next slide, please. So what we do not know yet is what happens if we introduce lunar dust into that ISS type dust that will also be present in a lunar habitat. And this is what we're proposing to do in the future. Next slide, please. So some potential impacts that we thought of about lunar dust infiltrating ISS type dust is microbial inhibition and or altered moisture absorption of the dust. So the composition affects microbial communities, as I mentioned before. So the introduction of lunar dust may destroy weaker microbes and allow stronger ones to survive, creating a situation where it goes from a high diversity or lots of different microbes to where one or two dominate. And also the lunar dust might affect the moisture uptake of the dust. And this is important because if moisture is absorbed into the dust, that moisture is now available for the microbes to use and grow. Next slide, please. So just to sum up, ISS dust can sustain significant fungal growth in elevated relative humidity conditions, and this type of dust will be found in lunar habitats. Um, and we need to understand how lunar dust may impact the dust and microbiome of a lunar habitat. And this is important for astronaut health and habitat integrity and planetary protection as well. Next slide, please. And I just wanted to thank NASA and the Space Biology Grant and the Ohio State Environmental Science Graduate Program for funding this. Also our collaborators, Dr. Myra Meyer from NASA Glenn and Dr. John Horak from Ohio State, and my principal investigator, Dr. Karen Dana Miller, and our amazing indoor environmental quality lab. And that's all I have. Thank you, guys.
Thank you, Nick. There were a couple of questions. There was some questions, but I believe Jim Bryan had taken care of them. Uh, Basically, what is the normal humidity on ISS or an Apollo spacecraft? And he said 60% is typical. Jim responded 60% is the typical ISS humidity, but can vary locally if the astronauts are exercising, or if they're in a what they call the hygiene area, such as the where they wash off at the end of the day.